Today, once again, in the third day of this national seminar, Nurturing Science through DBT STAR scheme, organized by Uday Pratap Autonomous College. The theme of the national seminar is Recent Trends in Zoological Sciences. And I'm extremely delighted to hear and see one of my students who is still associated with his alma mater and working as a scientist at Kentucky in the Tobacco Research and Development Institute, United States. Dr. Sanjay has brought laurels for our college, Uday Pratap Autonomous College. He did his graduation as well as post-graduation from Uday Pratap Autonomous College. And I'm really very thankful to him that he accepted my invitation and he has joined today to deliberate on a talk how to catch a right fish. I don't know whether he'll be talking about fish or whether he'll be talking about other animals or plants, but I know that it will be an interdisciplinary talk. He's working on tobacco plants, and I'm really thankful to him. He'll deliberate on lessons from genomics. He has been working on genomics since last several years. And let me also tell you that when I just joined, he was in BSc third year. So one of my first batch of students, and he dared to ask me my introduction in the class. And that day I thought that he might be a very naughty person in the class. But when I came to know him, he was really very studious and he qualified NET and JRF, worked at JC Bose Institute, and then the journey started for him and he has reached such a great height. So thank you so much, Dr. Sanjay, for joining us today and making our seminar a really interesting and enjoyable one. जो टॉपिक आपने रखा है वही देख के लग रहा है कि ये किस तरह से जो दर्शक हैं को बाइंड करेगा, क्योंकि अभी टॉपिक से कुछ समझ में नहीं आ रहा है कि हम क्या सुनने जा रहे हैं और ये लेक्चर लग रहा है कि बहुत ही इंटरेस्टिंग और बहुत ही अच्छा होगा। मैं डॉक्टर अश्विनी निगम, ऑर्गेनाइजिंग सेक्रेटरी ऑफ दिस सेमिनार, से भी बोलूंगी कि आप आपसे एक बार रूबरू हो और आपको इंट्रोड्यूस करें, वेलकम करें।
Welcome, sir. And as Dr. Tumul ma'am said, that this topic is seemingly very interesting because we are assuming that this topic would be a type of means lesson from genomics. What would be means topic from any related with the life science or part of the life science, or itself related to fish or related to any technique like fluorescence technique. So we will be definitely benefited from this lecture. And sir, you can start now.
Okay. Thank you, Tumul ma'am and Dr. Ashwini for the kind introduction. It is my privilege to be here with my roots. Actually, EP College, I consider it as my academic roots. And thanks for invitation. So I am going to talk about how to catch a right fish, lesson from genomics. So let's start from here. So actually, I use the word fish only because I believe there is a special paper fishery in the Department of Zoology in MSc. So fish here stand for a gene. And if genomics is a sea or ocean, so genes are a very tiny fish and you need to catch that right fish, a right gene, which is important for your research at whatever field you are working. So today we will talk about all this talk can be categorized in three different sections. So one will be the background, where I talk about that how the bioinformatics come into the field. And in second part, I will talk about the necessity. So why it is essential to use all those tools. And third part will be the real life examples. So here I will give some examples that how the group I work with use that tools to explore the genomics and catch the right fish or right genes which regulate the pathway they are working with. So most of us, can you see this picture? Yes, now you can
Yes,
see.
Yes,
beautiful
sir.
picture.
So yeah, so if you see the
Beautiful
picture,
picture of a
I
bridge.
think, yeah, most of know this bridge. This is the Malvi bridge near the Kasi station. So that bridge has very interesting story, very interesting history. So before the 1900, around 1885, there was a single bridge which were used by the rail or train and the land traffic like bus and whatever the available at that time. And in 1885, there was 10 or 12 trains used to cross the Ganga from the Chandauli side to the Varanasi side. So it was used to like that way that you have to wait until the train cross, and when the train cross, the other traffic can cross. So when the only 10 or 12 trains are crossing through the river, it is easy. But by the 1925, and it the numbers goes up by 30, then 50, and I don't know how many trains currently are passing through this bridge right now. So in 1925, the administration realized that this is not convenient to wait for maybe 50 trains in a day. So they decided to build a bridge in such a way that both train and the other traffic can pass through the same way. So as they say that necessity is the mother of inventions. So more you need, more you tools have to use to make your life easy. This is another example that how technology can, change our lives. So sufficiently advanced technology is equivalent to the magic. So everything or anything is magic unless we don't know the technology behind that one. So if you go to the first pictures, that is the Stonehenge from the England, so you can see that huge builders are putting in the circles. So definitely it was a miracle when it was built, but right now we have the sufficient tools and techniques which can build this all those structures maybe within a minute. So now the miracle has gone because we have the more advanced technology. The same way, if you take the example of Great Wall of China or the Malibu Viaducts of the France, so Great Wall of China is a structure which you can see from the sky, right? The same way, if you go to this bridge of the Millau Viaducts of France, you can see the clouds from the bridge. So all these miracles changed by the techniques. So let's give me let me give you another example here. You can see the three different sections. The first one is the polyacrylamide gels. I think the slides are not changing. Yeah, now it is changing. Sorry for
It's changing, Dr. It's
little
okay,
bit.
fine.
So
Little bit
the
slow.
yes,
You keep moving on the YouTube, it will be a little bit slow, okay?
okay, I got
Where
it.
you are watching on YouTube, it is a little bit slow, but here on
Okay,
this platform, it is moving, okay?
okay,
You
thank
go
you.
ahead.
Yeah.
Oh,
So you can, in 1980s, when my PhD supervisor was used to sequence a gene, so they used to run a huge polyacrylamide gels. So, and then they used to expose it to the X-ray film and then develop. So all this sequencing process of even a single gene used to take a month. When my PhD supervisor was using his PhD, and then he was doing his gene sequencing, they used to get the information in a chromatograph form, and then they save those data in the floppy drive. Nowadays, when we are doing the sequencing things, the file size and the sequence information is so huge that you cannot use a floppy drive to save that one. Even you cannot save those enough files in your computer. So for that one, either you need a cloud storage or a hard drive, external hard drive of huge capacity. So that's what that is the way actually last 30-40 years how the techniques has changed our whole perspective that we deal with the sequencing information right now. So let's talk about the bioinformatics and its application. So in 1970s, the people coined the term bioinformatics, which are defined as the idea of informatic process in the biotech systems. So there are several components of bioinformatics like the technology, algorithm, database, analysis, and visuals. And for every aspect, you need a specific tools and techniques to develop those, or even the language are different for that one. And if you go to the right part of these slides, so I'm talking about the different section or subsection which are utilizing these tools for their progress, like homology modelings of the proteins or phylogenetic analysis to get the evolutionary idea, the functional annotation or genome annotation to get the idea of a function of genes, or system biology, or omic studies like the genomics, proteomics, transcriptomics, and metabolomics. So all these things are possible only because of these tools which are developed by the bioinformaticians. And we as a I am basically a plant biologist. So as a plant biologist, this is the same true for the animal sciences also. So all those people are utilizing those tools to speed up or catch the right fish or right gene of their interest. So just for example, how the huge data is getting generated and how these tools are getting utilized by the different groups for their purpose, so that you can easily see those data and make a conclusion out of that. So for example, if you go to the left side, there is circle which is called as a circus plot. So circle plot has the different small circles and different the small pies. Like if you go to the one by one, like SB1, SB2, SB3, SB7, SB9, SB10, all these show the sorghum bicolor. That's a plant. And 1, 2, 3, 4, 5, whatever the number is, they represent the chromosome numbers. And then at every chromosome number, the scientists have plotted the information related to the transposition, exons, introns, or retrotransposition. So you can imagine that if you go one by one to visualize all these informations, it is impossible. But you put all this information through a bioinformatic tools, it is very easy to read those information. The same way, if you go to the middle or the last section of the slides, people have mapped the homologous part or similar part of chromosome of the brachypodium to the chromosome of sorghum, chromosome number 1, chromosome number 2, and 10. So these all sequencing information are so easily visualizable and readable only because people are using these tools which are developed by some bioinformatician. So the information is important, but the proper utilization is very important to catch the right genes. So current motto of the scientists are the sequence them all. I think in 2020, the Earth Biogenome projects make a aim to produce the reference genome for all eukaryotic species within the next decades. So if you go to the plants have the most threatened species than other targeted genomes, while the second most species are coming from plants after the invertebrates. So how come, it is possible to sequence most of the genome by the next decades? It sounds so ambitious, right? So here is the answer. For every passing year from the 2001 to 2021, the cost per raw megabase of DNA sequences is going down drastically from maybe 100,000 to less than $0.5, right? If you go to the right side, in 2001, the cost per human genome was more than 100,000. But right now, in 2020, it is coming below the $1,000. So you can imagine how it is easy to sequence a genome. So for example, if you decided to sequence your genome right now, it will cost less than 70,000 INR. So that's why people are talking about the personalized medicine and personalized cure and cancer and this and that, only because the sequencing cost is very, very low. This is another representation of the similar kind of data. So in 2003, the Human Genome Project cost more than 300,000. But in 2040, it was around 1,000. In maybe in 2020 to 21, it is less than 1,000. So things are getting cheaper because technology is going up and we are getting huge amount of data. So how the India stand in the bioinformatics field? So here in the left side, if you go to the plot from 2000 to 2018, the number of publication per year has gone from the 8 to 1600. And India is among the top 4 countries which are generating the data and databases every year. So there are huge number of scientists are involved, are using these tools in India too. So in this slide, I'm going to talk about how all plants are not equal. Equal in the sense of the genomic size or gene numbers. So here, this violin plot is showing the frequency and range of genome size in different classification uh, plant section, you can say. It's like in the left side, there are bryophytes and the lygophytes. They are the lower plants, and on right side, they are the higher plants. So, for example, if you go to the monocots, that is represented by here by the pink violin, the genome size varies from maybe somewhere to the 50 GB to the 150. So even in the same class of the plants, the genome size varies a lot. In this graph, I'm showing that how the genome size and gene number varies in the plants. Remember that in genomics, it doesn't matter whether I'm talking about the plants or animals. The basic concept is same. Like in plants, if the genome size varies a lot, that is true for the animals also. So here, if you plot the genome size versus the gene number, and if you guys know the Arabidopsis, so Arabidopsis genome size is around 150 MB, and gene number is less than 30,000. In the second part, if you go to the GMS, that is a corn, so it is around 100 or 200, more than 2000 MB, while gene number is 40,000. If you go to the soybean, the genome size is around 1,000 and gene number is 50,000. So I want to say that genome
Dr.
size.
Sanjay, Dr.
Yes,
Sanjay,
ma'am. Yes, ma'am.
Dr. Sanjay, can you hear me? Yes. Just pick that one, जहाँ पे होगा stop screen वाला एक border है. Can you see that border in your graph itself?
Yes, ma'am.
Okay, fine. Move ahead.
Okay. So just want to say that the number of gene and genome size, they varies a lot across the plant or animal. So what will you do with all this information? Say you sequence the all kind of plants, all kind of animals, and whatever you want. So there are two methods. One, collect all information and just store them somewhere. Or the second part is collect those information, clean them, and utilize them as soon as possible. So that's why the left side is always lesser than the right side. So only sequence or only informations are not important. How fast you process those information and how fast you utilize those information is important. So since I'm a plant biologist, again, I'm reminding because most of my examples are coming from the plants. But still here, I'm going to talk about the how plant scientists are utilizing the bioinformatics tools. So this circle can be classified into 4 different parts, or you can say the 4 different fields which are utilizing these tools. The one is genotypings, or phenotypings, or breedings, and every sector's the plots show the cost, data, point efficiency, and type efficiency. And you can say that as more information and more computing power are coming, the time efficiency and the cost data point efficiency is going up. For example, if you go to the genotyping, I mean, few years back, maybe 1 decade or 2 decades ago, people used to use the PCR-based marker for the genotyping, which was the low throughput, costly, laborious. Although it was highly flexible, but it takes lots of time. But by the time the new techniques introduced and new tools came in the market, in the fields, and now people are using the next generation sequencing, which is high throughput, very low cost, very, I mean, per sample is very easy to process them, and you are getting more and more information. So all these tools are making us to catch those right information, whether it is gene or protein or whatever you are looking for, so easily accessible. So just guess or imagine that what will happen if all these tools are not available. So how will you take the sequence information and get the gene function or functional associations of those genes to a pathway? So it will take lots and lots of time, right? So let me give you some examples which we are using in our labs. So how we use those tools are those concepts to catch the right fish or right genes. So mostly in the lab I work in, they are using a metabolic pathway which produce some anti-cancerous drugs in plants. So main goal is to find the regulators or transcription factors which regulate those genes, so that they can increase the efficiency of those genes, so that the plants can produce more anti-cancerous drugs. So metabolic pathways are set of actions or interaction between genes and their products that results in the formation or change of some components of the systems. And regulators which regulate the different genes, and any metabolic pathway can be regulated on the translational level or post-translational level or transcription or post-translation. I mean, there are so many levels which a pathway can regulate. So you can imagine the complexity of a pathway and their regulation and regulation after regulation, right? So for example, if there is a transcription regulation, a gene, how many transcripts are getting created in the systems, and then there is another microRNA, or people are using RNA. I mean, there are so many ways that people can regulate a metabolic pathway. So model system in the lab I'm work is a Catharanthus roseus. I work with the tobacco also, but today I'm going to talk about mostly Catharanthus roseus. That is a very beautiful ornamental plants. And people, I mean, I think in India, people say Sadabahar or something like that. People use in their gardens and some other places. And this plant is very important for the producing the anti-cancerous drugs, and those compounds are vincristine and vinblastins. And the pathway which produce these two metabolites are the terpenoid indole alkaloid pathway, or TIA pathway. So this plant is not merely beautiful plant, but very valuable medicinal plants. So how we use the techniques to identify the regulators of TIA pathway? So today I'm going to talk about the 3 different main way we identify the regulators. The first one can be the homologous transcription factor, which regulate the structurally similar or diverse specialized metabolites in different plant species. So in simple way, you can say that if, for example, if somebody is speaking Bhojpuri, so you can imagine that person may belongs to the either Eastern UP or Western Bihar, I mean, you can, I mean, it may or may not be that, but there is high chances that that person belongs to some region of that part, right? The same way, if some transcription factor is regulating a metabolic pathway in a say in a soybean plants, so you can imagine, you can make a smart case that it can be involved in the similar kind of pathway in some maybe corn or some other plants, right? The same the second way we would use the co-expression analysis. So here assumption is that the the transcription factors which regulate a metabolic or developmental pathway must similar or may similar the show the similar spatiotemporal expression pattern as the pathway genes they regulate. And third one is the cis regulatory motifs which are present in the promoter, and every transcription factor uh, regulate or bind with the specific cis elements. So if, and it is known that if some cis elements are present in promoters of a gene, so some specific kind of transcription factor may regulate that genes. So let's start from the co-expression networks and gene function prediction. So here you can get your expression data from the various sources like the different time point, different treatments, different tissues of the plant or animal. And then you take those expression data to make a network construction or module identification. And then finally you can go for the enrichment analysis or finding the hub genes. And finally you pinpoint more than maybe 2, 4, or 5 genes, and then go for the functional identification of those genes. So these gene co-expression networks can be used for the various purposes like the candidate disease gene prioritization or functional gene annotations or identification of regulatory gene. So in this paper, we identify a kinase which regulate the TIA pathway genes, right? So kinases are the post-translational regulators which phosphorylate a gene or transcription factors, and finally which leads to the suppression or induction of the expression of that genes. So here, if you go to the left side, that is a heat map where the yellow color show the higher correlation, and the green one show the less correlation. And here you can see that a map KK is highly correlated with the pathway genes, and we guess that kinase may involve in the regulation of TIA pathway genes, and then we functionally identify that kinase and prove with different experiments that yes, that kinase is involved in the TIA regulations. The second second way I talk about the cis-secting elements. So we know that the promoters are located in the upstream of any gene. Mostly, I mean, not necessarily, but mostly they are located in the upstream of the genes. And cis-acting elements are the small stretches, like maybe 4 or 6 base pair longs, which can be recognized by a specific group, a specific family of transcription factors. So, and every specific transcription factor bind with the given specific cis-acting elements. So that is kind of a smart guess that, I mean, as I say, that if somebody is speaking some special dialect, a special way of speaking, you can smart guess, oh, that person may belongs to this region or this part of the world. So that is not necessarily every time works, but I mean, most of the time it works. And then you utilize those cis-acting elements, the transacting factors and promoters, and if you finally go for the gene function. There are several databases, several tools like NuPlace, Jasper, Transfect we use on regular basis to identify those genes. So using these techniques, we recently identify a transcription factor from the Catharanthus, which is called as GATA1. So initial analysis show that in the Vendolin pathway, the specific kind of cis-secting elements are present in several genes like T16H2, 16OMT, T3O, T3, I mean several genes. And then we think that GATA may be involved in the TIA regulations. And finally, with the different, characterization techniques, we prove that yes, GATA is involved in that specific pathway. So next question you can ask, whether I can use the same techniques or same tools in other plants or other animals or some other systems or not. So my answer will be yes, you can use. And we use the same things or similar kind of approaches to identify a transcription factor from the tobacco. And we file an international patent, which is already approved in some parts of the world, like India, Bangladesh, and some other parts. It is still under process, but yes, you can use the similar kind of techniques to identify and to the genes or fish the right genes, catch the right genes. Now, next question is, next approach can be the gene clustering approach. So gene clustering approach is, if a similar kind of genes are located in the vicinity in the genome, so they may involve in the regulation of similar kind of pathway. It is like, say for example, it is kind of hostler kind of, you can imagine this. Say if 2 or 3 hostlers are sitting together, they may involve in some kind of naughty things, right? So it is something like this approach may not work. There may be some exceptions, but it works in most cases. For example, if I took the ORCA3 was a transcription factor, which are way before that, way before the genome sequence of Catharanthus was available, was identified. And when the genome sequence of Catharanthus available, maybe 5-6 years, and then people analyzed the part where ORCA3 was located, and they identified that similar kind of genes like ORCA2, 6, 4, and 5 were also located in the same region. And in our lab, we identified the ORCA4, 5, 6, and experimentally validated that these genes are involved in the TI regulation, but ORCA2 is not involved in TI regulation. So as I said, 3-4 hostlers are sitting together, may do some naughty thing, but there are always some exceptions. So next thing you can ask, okay, you guys identify the ORCA3, 4, 5, whether similar kind of clusters are present in some other plants also, or whether they are involved in this similar secondary metabolite production regulation or not. So my answer is yes. Just like the Catharanthus, the similar kind of clusters are present in the giant milkweed, jasmines, tomato, potato, tobacco, several plants. And it is already known that in Catharanthus, tomato, potato, and tobacco, they are involved in secondary metabolic regulation. So this is the way things work. So you get the information, you have some hypothesis, make a smart guess, go for the identification. So instead of going through the 100 subgenes, you choose 3, 4, 5 genes, go for the characterization. It is so easy. The same approach, we also identify the BIS3. So BIS1 and BIS2 was reported before, and when genome was available, we reanalyzed that part where BIS1 and BIS2 was found, and we identify another gene BIS3, and we recharacterize those in the different plants, and we find that yes, they are involved in the TIA pathway. So, and these kind of clusters are present in the Arabidopsis, in the Calotropis, and Gelsemium. So the Calotropis and Gelsemium is still less explored plants, but in Arabidopsis, they are also involved in the metabolic regulation. So that is the way you get the information, and then you process them, you minimize the candidate numbers, and go for the characterization. So in summary, I want to say that bioinformatic tools and methods are mandatory to organize the use data and extend our ability to analyze the complex biological systems in very fast manner. So Omic has allowed us to come to terms with the vast amount of biological data which are being generated by the sequencing projects, and has made considerable progress in biology, both in plant and animal systems. So in plant biology, these bioinformatic tools are very helpful in improving the quality of traditional medicinal plants. You can improve crop nutrition quality, the single gene analysis, the sequence similarity modeling. I mean, there are so many ways you can use these tools to improve our you make it fast. So final question is information and intent. So is it true that information is equally proportional to speed? No, it is not true. Intent is very important. So techniques and resources are useless without the willpower. So let's go to the example for the Malviya bridge versus the bridge which is right now located in the Ramnagar. So if you go to the history, it takes 3 years to make the whole Malviya bridge in 1948 or something, plus minus 1 year. But in the Ramnagars, it took more than 12 years to just construct the skeleton. So I cannot say that the techniques or tools are lesser value in the maybe 2015 or something in comparison to 1948. But what was lacking? The intent. So information and intent both are important to make you successful. I'm thankful to my current supervisor, Professor Ling Yuan and Dr. Sitakant Patnaik, and most of these works were done by Dr. Patra, Dr. Liu, and Dr. Paul. I'm again very thankful to Tumul ma'am for inviting, and it was great get together for me to be with all of you, and I'm very, very, very feeling very good. Thank you, everyone. Thanks for your concentration, and I'm ready for any questions. I have some questions online too, so I will try to answer. सो एनपी सिंह सर ऑनलाइन है, तो सर थैंक यू, मतलब आप हमारे वार्डन भी रहे हैं और टीचर भी रहे हैं, इससे बड़ा हमारे लिए कुछ भी नहीं हो सकता है। प्रणाम आपको। अंजनी हमारे बैचमेट रहे हैं और अभी वो प्रोफेसर हैं आत्माराम कॉलेज दिल्ली में। और कोई क्वेश्चन है तो आप लोग पूछ सकते हो।
Well, Dr. Sanjay, I can see there are some kind of comments on the YouTube. Dr. Paya has written, similar kind of approaches can be used in important crops which impart resistance against all kinds of biotic and abiotic stresses. And
yes, ma'am. That is true.
Yes. So it's really great going in this seminar that we had today, such an interesting lecture, and from your topic we were feeling like it's something going to be very mysterious or what. So what are we going to hear from you? And you have really tried to correlate the plant genomics with the animal genomics and putting there a word or a term fish. It really brought the platform to an interdisciplinary approach, and this is the need of the hour. And you have very well given those examples which are really very important, like you gave certain examples of maize and other plants, so that can be utilized, those data can be utilized for working with animals, because when we talk about biotechnology, genomics, proteomics, it is really very difficult to start working on the animal model system, because you never know what will be the kind of complications or what will be the outcome. But plants are easy to handle, easy to cultivate, then easy to do some kind of genetic engineering, and you can catch the right fish, what you call the right gene from the plant, and then identify that gene, and then how it works, how it is regulated, and then you get a pathway from which you can then incorporate your work with some kind of bioinformatic tool on the animal system, and it becomes easy. Another thing is that it is very important from the comparative aspect, because when we see the evolutionary significance of or phylogenetic relationships then also these kind of work helps us improve the understanding of science,
Yes.
helps us improve our quality of research. And the best part of your presentation was like, uh, it showed how much you are aware of the past and also about the future, and you have compared with such beautiful example like the Maria Bridge and the other bridge. So this is the kind of goodness we really wanted. And when I used to teach you, I had seen something in you that like you will reach a level जहां आप हमसे ऊपर होगे, और इस बात से
Definitely.
रियली हमें गर्व है। और आपने इस इंस्टीट्यूशन के लिए, उदय प्रताप कॉलेज के लिए और उदय प्रताप जूदेव जी के लिए आपने काफी कुछ किया है, आपने इस पटल पे लाया सारे चीजों को, आप इस पे पहुंचे, ये देखकर बहुत ही अच्छा लग रहा है। और एनपी सिंह सर हमारे साथ जुड़े हैं। मैं
Yes.
चाह रही सर को भी जोड़ा जाए, डॉक्टर अश्विनी, कैन वी डू दैट?
Yes, ma'am. If you could provide link, guest link to
यस,
him.
आई एम ट्राइंग।
If you just forward that link to Dr. MP Singh sir.
तो अश्विनी फिलहाल आप बात करें थोड़ा डॉक्टर संजय से,
Oh, okay.
मैं देख
Yeah,
रही
yeah,
हूँ।
yeah, ma'am. I'm interested to talk with. Okay, thank you, Dr. Sanjay, for delivering
So
such an interesting lecture on the topic how to catch a right fish lesson from genomics in a very simple and beautiful way. By this lecture, you have also changed our mind to view the word fish. Actually, in our classes, we told a fish like a living animal. Okay, and the word fish, it is also representing the catching anything out of the big ocean. And you have also correlated with it in a very nice manner with different types of omics like genomics, proteomics, and metabolomics. And yes, catching, identifying, and characterizing any gene out of the whole genome is very similar to a finding and catching a living fish out of
yes, sir.
ocean also. And I am
Yes,
very much
you are
agree
so
with
right.
you, sir. And you also presented different techniques with basics starting from the also the evolution of the technique to characterize and study the genes and related molecular biology and functional genomics of the human and life itself. And we are very much just thankful to you for such a nice deliberation and such a nice presentation, sir. And one question from myself is that you are working on, sir. Sorry, I could not catch your topic of research.
So actually, we are working with two plants. So one is the catharanthus, and
Okay, yes.
another one is tobacco plant. So
Okay.
catharanthus, actually, you can, there is a contradiction, you can understand that catharanthus produce the anti-cancerous drugs, right?
Yes.
And the tobacco cause cancer. So I mean, in cancer we are trying to produce more anti-cancerous drugs, and in tobacco we want to reduce. We are working in the harm reduction. That how you reduce those cancerous products in the tobacco
Yeah,
plants.
yeah.
So it can be more. I mean, how to say that, human friendly or more healthy compared to the wild, right?
Yeah, yeah, yeah. Yes, sir. Actually, after listening your presentation, actually I have worked on the fish immunity, and as a fish biologist and man of fishery, I am very much interested to see the effect of these anti-tumor effect having plants, or you can say anti-tumor effect of these plant extract on the fish also, because in fishes there are several types of tumor and cancers also, and
Yeah.
such type of diseases are well diagnosed and researches and still going on to treat in human. But in fish, to increase the population, I think such type of interventive experiments and integrative experiments are needed to just cope up with these diseases in fish, because in this era of environmental pollution, aquatic pollution, there are certain different types of mutagens which are also affecting the health of the fish. by just inducing and promoting tumors and cancers, which are well neglected area in the part of the life science. And I'm I will be interested to see the effect of these plants itself, tobacco, and it is very much easily available in the market.
Yes, sir.
Can we do, sir, in this manner? How means if you I am very much interested to see the effect of the extract of plant. If you could just direct us to just plan such type of research, I will be thankful to you, sir.
So
Can you please, sir?
you want to see the extract of plants on the fish?
Fish, yes, having any type of tumor or cancer. It is very normal in fishes nowadays.
So I mean, there are so many way people are using I mean, this is very basic research people are using. Like you feed the like you feed the like how to do, like say you want to somebody talking about the biotic stress. So what people will do, say
Hmm.
you overexpress a gene in
Yeah.
plants
Hmm.
and then feed to some insect. Same way
Okay.
you can extract those plants and then feed it to the fish and then see whether they are developing the cancer or not.
Okay. 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 Yes. Yes. Means, sir, another plant on which you have worked at Catharanthus, if I'm not
Yeah,
wrong, sir.
yeah.
Yeah.
Catharanthus,
That Catharanthus.
I think Catharanthus plant you can find in India too, because there are
Yeah.
several labs which are working on the Catharanthus from India too. Like one group from the CMAP, Lucknow, and then there is
Okay. Okay.
another group from the Delhi. So I mean, for a
Hmm.
long time, people from India are working on that plant.
Okay, means for that I have to just make a preparation or extract of that catharanthus and then mix it with the feed of that fish, or we may also inject, sir, if we find any pure
Okay.
uh,
Okay.
compound to treat.
Let me put it this way. I am
Yes,
not the
yes.
right person how to treat the fish. There are
No,
so
no, no,
many.
no, sir. No, no, I'm not talking in that way, sir. I'm just
So there are so many nice labs in the BHU. I think one of my colleague, Dr. Vinay Kumar Singh, was used to work in the lab. I think Dr. Bichan sir. So they are
yeah,
really
yeah,
nice. So
yeah, yeah,
so
yes, sir.
they
I
exactly
have
know
worked
how
with
to
him.
how to deal with the fish. I can say
No,
that
no.
yes, that is doable. I mean, you can feed the either you feed the extract or the pure compound, and you can see the effect. But I'm not the right person to ask that how to treat the fish, right?
No, 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 sir. Actually, we have done such type of experiments by injecting the albumin into
Huh.
the intraperitoneal cavity of the fish to see the immunostimulant effect of that albumin extracted from the aloe vera. Uh, but I want just to know that such type of extracts from like containing wind blasting and wind twisting
Yeah.
in
So those compounds are available from the
Catharanthus.
Sigma. I mean, the Sigma
Yeah,
you
yeah.
can
That's
buy them, and I think you can feed them to see the effect of those compounds on the fish. Minu is
yeah.
online.
That is I am.
Minu was my batchmate, and she's a very really scientist.
Yeah, yeah. Okay. No, no. Actually, thank you, sir, for
Hello.
clearing my doubt.
Hello.
And now over to you, ma'am.
Yes, Dr. NP Singh sir has joined, and he will also
Good
speak
morning,
few
sir.
words.
Sir
We
Pranam.
have a nice experience about Dr. Sanjay when he was studying at this place. Dr. Ashwini, can you mute yourself?
Yeah, 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 ma'am.
Yes.
Sir Pranam.
सर बोले सर की आवाज आवाज नहीं आ रही है
हाँ जी, हाँ जी.
सर अनम्यूट है
नो मैम, अनम्यूट तो नहीं था, नॉर्मल था सर का, बट आवाज नहीं आ रही थी सर की।
माइक मोड पे है क्या?
नहीं सर, एग्जिट हो गए हैं, फिर से अभी शायद एंटर करें। आई एम वेटिंग जस्ट।
हाँ, तो डॉक्टर संजय रियली बहुत इंटरेस्टिंग था। और ये बायो इन्फॉर्मेटिक्स के जो टूल्स हैं, इससे कैसे हम क्रॉप इम्प्रूव कर सकते हैं, क्रॉप के क्वालिटी को, क्रॉप के प्रोडक्शन को, डिजीज रेजिस्टेंस को, या फिर क्रॉप इंटायर क्रॉप मैनेजमेंट में, ये सारी ही चीजें बहुत ही इंटरेस्टिंग थी। और रियली बहुत अच्छा लगा आपको सुन के आज। और मैं चाह रही हूँ कि एनपी सर भी जुड़ें, क्योंकि
हाँ, एनपी सिंह सर हमारे वार्डन रहे हैं।
हाँ,
मैं मतलब जो नहीं जानते हमको पता है। और सर के साथ बहुत सारी स्मृतियां जुड़ी हुई हैं हमारी। हम अभी भी मतलब याद करते हैं
संजय जी को बहुत
उनको।
बहुत बधाई।
धन्यवाद सर,
वेलकम संजय
प्रणाम।
जी।
प्रणाम सर,
प्रणाम
आप
प्रणाम।
कैसे हैं?
बहुत अच्छे हैं जी।
आपका
अच्छे।
आशीर्वाद है सर, और कुछ नहीं।
नहीं, आप, हमारा आशीर्वाद तो आपके साथ हमेशा है ही है। आप जो है, अब हम लोग शुरू करें मैडम, क्या बोल रहे हैं?
हाँ सर, आप बताएं। हाँ।
आज
सर आप बोलिए। हाँ।
फर्स्ट ऑफ ऑल आई लाइक टू वेलकम डॉक्टर संजय कुमार सिंह।
थैंक यू सर।
The talk given by the Dr. Sanjay Kumar Singh was excellent and very fruitful for our students. And this is very uh, interesting to know how the things which are related to our society and students. And the Sanjay Kumar Singh was the very excellent student, very intellectual during the study. और सबसे अच्छी बात है कि ये डे बाय डे इम्प्रूव करते गए,
आप
डे
लोग
बाय
का
डे
आशीर्वाद
इम्प्रूव
है सर
करते गए।
मेरा।
और आज इनको इस स्थिति में देख के सभी लोग, हम लोग बहुत प्रसन्न हैं।
धन्यवाद
तो
सर।
ये एक मोटिवेशन है डॉक्टर संजय सिंह का, यहाँ पे जो टॉक दे रहे हैं, ये सारे बच्चे यहीं से सीखें और आगे जाएं और इस ऊंचाई पे पहुंचे जहां आज संजय जी हैं। संजय जी ने हम लोगों को के इस इनविटेशन को एक्सेप्ट किया और अपना पूरा समय इसमें दिया है। और आगे भी हम लोग उम्मीद करेंगे कि डॉक्टर
नहीं,
संजय जी
ये मेरा
हम लोगों से
सौभाग्य है
जुड़े
सर कि आप
रहें
लोगों ने मुझे बुलाया। नहीं तो फिर मतलब
और आगे भी आप डीबीटी की एक्टिविटी में और सारी एक्टिविटी में हम लोगों के साथ रहें।
जी।
और जहां भी हम लोगों को जरूरत पड़ेगी, हम लोग आपको कॉल करेंगे और
जी।
आप इससे जुड़ेंगे तो हम लोगों का भी जो है सम्मान होगा। और डॉक्टर संजय जी के बारे में थोड़ा सा मैं बताना चाहता हूं पर्सनल भी, कि ये बीएससी में हमारे हॉस्टल में थे और डे बाय डे ये इम्प्रूव करते, इन्होंने अपने को हमेशा इम्प्रूव करने के लिए ही लगा रखा। इनकी एक्स्ट्रा करिकुलर एक्टिविटी या जितनी भी पढ़ाई से एक्टिविटी, ये सब कुछ इनका अपना है। इन्होंने सब कुछ अपने से क्रिएट किया है। इन्होंने कुछ पहले से लेके नहीं आए थे, लेकिन सब कुछ इन्होंने क्रिएट किया, यूपी कॉलेज से पाया और उसके बाद आगे बढ़ते गए और पाते चले गए। तो ये हमारे डीबीटी के बच्चों के लिए, जो बच्चे घबराते हैं कि हम कुछ नहीं कर सकते, थोड़ा कम कर सकते हैं, ऐसे बैकग्राउंड से हैं, संजय जी उनके लिए एक एग्जाम्पल है। और उम्मीद करेंगे कि हमारे और बच्चे भी इस तरह से अपने को एडजस्ट करेंगे, इस तरह से अपने को इम्प्रूव करेंगे कि वो संजय जी के पैरेलल खड़े हो सकें। इसी उम्मीद से कि सारे बच्चे इनसे मोटिवेट हों। और इस उम्मीद से मैं संजय जी को भी धन्यवाद देता हूँ, बधाई देता हूँ कि आपने इतना अच्छा टॉक यहाँ पे दिया है, जो हमारे बच्चों के लिए बहुत मोटिवेशन का काम करेगा। और आगे हम सारी उम्मीद करेंगे कि आप हम लोगों से जुड़ते रहें।
धन्यवाद।
मैं डॉक्टर तुमुल जी को धन्यवाद देना चाहता हूँ कि आपने संजय जी को बुला के और हमसे इंटरेक्ट करा के
धन्यवाद।
एक हमें अपनी अपॉर्चुनिटी दी कि हम संजय जी से फिर से जुड़ सके और कॉलेज के लिए इनका उपयोग कर सके।
धन्यवाद सर।
धन्यवाद। जय राजर्षि, जय भारत।
जय राजर्षि।
थैंक यू डॉक्टर एनपी सिंह सर। और रियली आज बहुत ही अच्छा एक दिन भी है काशी के लिए, और जहां तक मुझे लग रहा है कि शायद ये एक हिस्टोरिकल दिन है काशी वासियों के लिए। और आज ही का दिन हमारा एक तरह से ये भी हिस्टोरिकल हो गया कि हमारे एक स्टूडेंट जिन्होंने हमारे कॉलेज के लिए लॉरेल्स लाया है और कॉलेज का नाम रोशन किया है, आज हमारे साथ जुड़े हुए हैं। और मैं आपको बताऊं कि डॉक्टर संजय कुमार सिंह के इस जुड़ाव से आज लग रहा है कि शायद ये नेशनल कॉन्फ्रेंस, एक इंटरनेशनल नेशनल सेमिनार जो हमने कराया, ये एक इंटरनेशनल सेमिनार का रूप ले लिया है। और मैं अभी देख रही हूँ यूट्यूब पे एक कमेंट आया है, डॉक्टर मीनू केशरी,
मीनू हमारी
इन्होंने लिखा है, ये हार्टी कॉन्ग्रेचुलेशंस डॉक्टर संजय सिंह, फीलिंग प्राउड बीइंग योर बैचमेट, हार्टी थैंक्स टू डॉक्टर तुमुल मैडम फॉर दिस प्लेटफॉर्म, डॉक्टर मीनू केशरी, पोस्ट डॉक साइंटिस्ट, बोस स्टेट यूनिवर्सिटी यूएसए। तो ये सारी चीजें जो है इस मेंटरशिप प्रोग्राम में आप जुड़ती जा रही हैं, और मुझे लग रहा है कि आने वाले समय में इसी बार जो डीबीटी के तहत प्रोग्राम हो रहा है, आप लोग फिर से एक बार मेंटरशिप प्रोग्राम जब मैं कराऊंगी तो आप लोग ऑनलाइन प्लेटफॉर्म पे जुड़ेंगे, और उस दिन छोटे बच्चे होंगे, स्कूल के बच्चे होंगे, और वो जब आपको देखेंगे कि चंदौली से आया हुआ एक बंदा जो बिल्कुल अनभिज्ञ था साइंस के बारे में या अपने एम को लेके नहीं आया था, वो किस तरह से आज यहाँ तक पहुंच पाए हैं। और ये एक बहुत अच्छा एग्जाम्पल सेट होगा। तो मैं आपको फिर से इनवाइट करूंगी, आपका अगर आधा घंटा भी मिलता है तो इट इज वेरी इंपॉर्टेंट फॉर अस। अभी वहां पे रात है मुझे मालूम है, फिर भी संजय जुड़े हुए हैं हमारे साथ, और ये रियली इसको एक आपने अलग ही स्वरूप दे दिया मेरे सेमिनार को। थैंक यू सो मच, एंड थैंक यू एनपी सिंह सर फॉर ज्वाइनिंग अस एंड मोटिवेटिंग मी। थैंक यू सो मच।
धन्यवाद।
Thank you, madam.
Thank you, sir.
Thank you,
so we'll have our next session at 11 o'clock. So I think I should end now. We will stream again for the next session. Thanks to all.
धन्यवाद।
Thank you, Sanjay.
Thank you, ma'am.
So we all can leave now so that we can end the broadcast.
Yes.
Okay.